Guys, we're going to talk about DNA, which is the hereditary material and a complete code of life. It is made up of phosphates, sugars, and nitrogenous bases. And DNA appears in the cells in the shape of the chromosomes. Chromo means colored, soma means body. So these are some sort of color threads in the nuclei of the eukaryotic cells, whereas in prokaryotes, it's present in the cytoplasm. So on the basis of the arrangement of the chromosomes, living beings are divided into two groups. One is prokaryotes and one is eukaryote. As we see here in this diagram, so towards our left hand side we see a prokaryotic cell. So you would see here the DNA is freely present, there is no membrane outside this one, whereas towards your right hand side you would see a eukaryotic cell. So the DNA, which is packed in the chromosomes, is present in some visible entity in the middle, which we call it as nucleus. So there is a complete nucleus present in the eukaryotes, whereas prokaryotes, they lack the nucleus. Same way in the eukaryotes, you also see some organelles which are membrane bounded, like this guy, green color, chloroplast, and this mitochondria. Both of them, they also contain some additional threads of DNA. So those, we name them as chloroplast chromosomes or mitochondrial chromosomes. Okay, so here is in this diagram, we also see the arrangement of the chromosomes. So as we see here, there is a nucleus and there are the chromosomes, and then we can see there is a single chromosome which is enlarged here. And then if you take this chromosome and start following it, you would see there is a thread which is wound on some protein molecules, which are actually, we call them as histones. So we keep on moving, and then till the end we find there is some double helical entity. There are two helices which are wound on each other, and they are made up of some things like A's and T's and G's and C's. So let's look into what these things are. Okay, so here are the guys which gave us this fancy structure of the DNA, Watson and Crick. So they were chemists. Actually, they were chemistry people who found the structure of this DNA in 1953. As you can see here, they are standing with their structure, which is made up of some clamps and some strands, which probably they are using from their laboratory. So as Watson and Crick indicated, the DNA is composed of nitrogenous bases, phosphates molecules, and sugars. Here we see on the top we have those nitrogenous bases. On the top there are the big ones, which we call them as purines, and on the bottom there are the small ones, which we call them as pyrimidines. So they are named as adenine and guanine, which are purines, and then we have pyrimidines, which are cytosine, thymine, and uracil. Thymine and cytosines, they replace each other in another molecule, RNA. In RNA there is uracil, but in DNA there is thymine. If you look towards the bottom of the slide, here we have a nucleoside. So nucleoside is when a sugar molecule, which is actually a 5-carbon sugar, ribose here, so that gets attached to a nitrogenous base, so it makes a nucleoside structure right here. Now when this nucleoside gets attached with a phosphate group, so we call it as a nucleotide. So here is a nucleotide, and towards your right there is a deoxyribose sugar present, which is there in the DNA. So if you look into this position number 2, the second carbon, this is position number 1, right, the carbon right here, and then we start from here and we start moving towards left. So this blue color hydrogen ion indicates that in this position there is no oxygen, so we call this sugar as deoxyribose. So that is the structure which is present in the DNA, whereas the structures which are present towards your left, they are common in the RNA. Okay guys, so now we move on to the structures of DNA. As we see here, there is a double helical string that they are wrapped around each other. So there is one red color string and there is one green color string. What are these basically? These are two strands of DNA, and if you look here, we see there are some numbers written here, 5 dash, we call it is prime, and then we have another number 3 prime. Here is 5 prime, here is 3 prime. So these are actually the directions of the two strands of DNA. On the back side, we call this structure as the backbone. So the backbone of the DNA contains sugar, phosphate, both of them as you look into. and nitrogenous bases are in the middle, right? So DNA has a backbones, and then they have the bases in the middle. As far as the directionality of the DNA is concerned, so the strands, they run from 5 prime to 3 prime end. So if you look into those sugars, this is your carbon number 5, this is 4, 3, 2 and 1. So nitrogenous bases, they are attached on position 1, whereas the incoming phosphates, they are attached to position number 3 of this one. And there is the one side that has 5 prime phosphate which is exposed. So this is the 5 prime end of the DNA, one of the strands of the DNA, and this is the 3 prime end, right? So whereas in computational biology or in computing, we can define DNA is a character string derived from 4 variables, which are A, T, G, and C. Down below, you can see here the structure of one gene. This format is called as a FASTA format. So you can see this one single strand of the DNA. There are some T, T, G, C, C. What are those? So basically in the DNA, there are just these 4 variables, A, T's, G's, and C's. So these are those variables which are represented. So in this way, we represent the DNA in our databases. So this side is obviously the 5 prime side, and this side is the 3 prime end. So DNA runs from 5 to 3 prime end.